This is the Professor, CBMG PhD. And today is Tuesday, May 13th, 2025. It is 7:26 and 38 seconds, p.m. Eastern Standard Time. And today's research, double-stranded DNA. So let's jump to it here. The best known set of structural proteins belongs to the histone family. These proteins are found in all organisms. There are thought to be some of the earliest proteins to appear during evolution. When associated with DNA, they form spools called nucleosomes, core particles, similar to those used to store thread, string, wire, and the like. This is level 2 of DNA organization in eukaryotic cells. These spools are composed of 2 copies, each of 4 different histones, named H2A, H2B, H3, and H4. The histones contain many positively charged amino acids, which attract them to the negatively charged backbone of DNA in a way that is largely sequence independent. The double-stranded DNA molecule is wrapped around a histone spool approximately 1.7 times 167 base pairs. This spool plus the short stretched 20 to 50 base pairs of DNA is called linker DNA that lies between spools is called a nucleosome. A linear arrangement of several nucleosomes forms a structure that looks like a string of beads. The addition of a linker histone, either H1 or H5, pins the DNA to the core particle, resulting in a structure called a chromatosome. Similar structures are found in prokaryotic cells, where the DNA is wrapped around a different set of histone protein spools. Wrapping DNA in this fashion causes the DNA double helical strand to become shorter and thicker. The length is reduced by approximately 7-fold, and the width increases from 2 nanometer to about 11 nanometer. These beads on the string structures also contain proteins, in addition to histones, in both prokaryotes and eukaryotes. Does wrapping DNA around the spool have any negative consequences? Remember that the goal is to compact DNA without compromising a cell's ability to access the genetic information. Because so much of the DNA comes into contact with the spool, most of it is inaccessible to other DNA-binding proteins, thereby negating the beneficial effects of these spools. In eukaryotes, DNA can be partially unwrapped from the nucleosome by members of the SWI-SNF family of proteins. These proteins use ATP, adenosine triphosphate energy, to move the core particle a short distance along the DNA, thereby freeing up any base pair sequence that may have been buried in the core particle. At least 2 other families of proteins participate in this type of chromatin remodeling as well. This video was created for educational purposes only. Codename Will, Professor Christopher Brunet, leader of research, scientist, ufologist, psychic, computer programmer, computer technician, philanthropist, writer, mathematician, artist, photographer, film producer, chef, animal, and nature lover. Passion, world peace. Hope you enjoyed listening. Until next time, have an excellent day. This is the Professor, CBMG PhD.